So today we're going to be learning about some topics in heredity, which include genotype versus phenotype, ratios, percentages, and a short intro to Punnett squares. So first let's take genotype versus phenotype. A genotype is an organism's genetic makeup or its alleles. And if you remember back from our previous lectures, you'll remember that alleles are copies of genes. They are the different varieties of genes, and they come in pairs. So it is the organism's pairs of alleles. And so when you think of genotype, think of gene in genotype as well as the word gene. They both have G-E-N. And some examples here are the two uppercase A's, the uppercase and lowercase a, and the two lowercase a's. Genotype is always expressed as the pair of alleles, and the alleles are represented by upper and lowercase letters. So the genotype will always be a pair of letters. The phenotype is an organism's physical appearance, so it's visible traits. And this is always a description, such as here we have a black mouse and a blue mouse. So these mice can be either black or white. That's the possible phenotypes. And in eye color we have blue eyes and brown eyes as the two possibilities for phenotype. Remember that trait is, in the mice, it's fur color, and in the eyes, the trait is eye color. But there are two different varieties here. And again, phenotype is how a trait looks or is expressed. And when you think of phenotype, think of the word physical. Both begin with PH. So phenotype is the physical appearance. And now we'll talk about homozygous versus heterozygous. And these two words, there are terms that are used to describe an organism's genotype. When we're talking about heterozygous and homozygous, we're talking about the phenotype. I'm sorry, the genotype. The genotype. And homozygous contains a word homo, which means it's the Greek word for the same. So homozygous means two identical alleles or two same alleles. For example, two uppercase B's and two lowercase b's. And those are dominant and recessive alleles. So homozygous can be either dominant or recessive. And heterozygous, the word hetero in Greek means different. So heterozygous organisms have two different alleles, represented here by the uppercase B and the lowercase b. So it's a combination of dominant and recessive alleles. And in this case, the phenotype is always going to be dominant, where in homozygous, when you have either two dominant alleles or two recessive alleles, the phenotype expressed there could be either dominant or recessive, depending on the allele combination. And in this example, we have a cross between a father and a mother who are both heterozygous, and in this example, the uppercase B's that represents the color brown, which is dominant. And the lowercase b represents green, which is a recessive. And in the offspring, we see the two uppercase b's. Those are homozygous dominant, and their appearance is brown. So their phenotype is brown. Their genotype is homozygous dominant, but their phenotype is brown. And then we have an offspring that has the two lowercase b's, which is homozygous recessive. The genotype is homozygous recessive, but the phenotype, its appearance, is green. And then we have two offspring that are heterozygous, and that's represented by an uppercase B and a lowercase b. Note that they have two different alleles. They have a combination of dominant and recessive alleles. Its genotype is heterozygous, but its phenotype is brown. It's showing the dominant phenotype. Because remember, anytime you see that dominant allele, the dominant phenotype is going to be expressed. Okay, now something we need to know about before we talk about Punnett squares is probability. And in particular, probability can be expressed in genetics as either ratios or percentages, depending on what the question's asking for. So probability is the likelihood that an event would happen. And in genetics, we're talking about the chance of an offspring having a particular genotype or phenotype. And the probability in genetics is either expressed as a ratio or percentage. In math, there are other ways to express probability, but in genetics, we express it in either ratio or percentage. Again, it depends on what the question's asking for. And a ratio is a comparison of two or more quantities or number, and it's to show how they relate in proportion to each other. So it's showing a proportion relationship. And for Punnett squares, a ratio is written with a colon separating it could be two or three numbers being separated by a colon. For example, a 3-to-1 ratio. The colon stands for 2. So a 3-to-1 ratio. And both genotype and phenotype can be expressed as a ratio in a Punnett square. And let's take that Punnett square that we had earlier and take a look at ratios. For the genotypes, we had three different genotypes. We had the homozygous dominant, the heterozygous, and the homozygous recessive genotypes. And they are occurring in a 1-to-2 to 1 ratio. So 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. And if we look at phenotype, we have, if we want to look at the ratio of brown to green offspring, we have a 3-to-1 ratio. We have 3 brown offspring and only 1 green offspring. And percentage is any proportion or share in a relation to the whole. And both genotype and phenotype can be expressed as percentages. And in the example of our Punnett square, if we're looking at the offspring here, the letters that are inside the boxes. And our genotypes for the offspring, the homozygous dominant, 25% are homozygous dominant. 50% are heterozygous, and 25% are homozygous recessive. For our phenotypes, 75% of offspring are brown, and 25% are green. Now I want to very briefly introduce you to Punnett squares. We're going to be doing a lot of Punnett square practice in the next few lessons in class, but just want to introduce you to what a Punnett square is. And a Punnett square is a tool that helps to show laws of probability and how it applies to genetics. And it's a chart, as you can see here, 4 squares. It's a chart that shows possible ways that alleles can combine in a genetic cross of 2 parents. So in a genetic cross, the combination of alleles that the parents can pass to offspring is based on probability. And using this Punnett square, we can use it to see the combinations of alleles in offspring and determine the probability of a particular outcome or result. You can use this Punnett square to determine the probability that an offspring will have a particular genotype or phenotype. And if you look at the top and on the side of the Punnett square, those are the 2 different parents and their alleles. So you have, at the top, you have one parent that is a homozygous recessive, and on the side you have a parent that is heterozygous. And each square inside this Punnett square represents the possibility for each offspring. So if there were 4 offspring, these would be the possibilities. But this is the possibility for, these are the different possibilities for genotype and phenotype and offspring. Okay, thank you very much, and hope you enjoyed the video, and see you again next time.